be posted pretty soon after the end of class. Also, I'm going to be putting the handout version of this PowerPoint online as well, if I didn't already. I think I might have already done that, actually. So you have two different things as a review for test one. You have review questions, which I've been posting, and I've updated those as of yesterday to include the nucleic acids. And today we're just going to talk about, just as an overview. If you have questions, stop me. If not, then we're just going to work through some examples, and I'll give you some ideas about what my expectations are. So we have 3 chapters on this test. The first one is that big overview of biology, and it's kind of a smorgasbord of different themes, so we'll be looking at that. Chapter 2 is basic chemistry, and chapter 3 is biochemistry, more or less. So here are the different topics we covered in chapter 1. We started with biodiversity. You watched a little video clip on the Foya Mountains and read about them. And I have some questions about that. And some of those questions relate to, can you predict where you would find the nearest species to those that are in the Foya Mountains? So let's talk about that a little bit. We actually have a map in this room, which is kind of nice. Where are the Foya Mountains? Where are they on the map? Andrea, can you find them? I'm going to put you on the spot. No. Okay. Do you know where, let's just start at the beginning. Do you know where Australia is? Okay, cool. So point to Australia on the map for us. Well, get up and walk over there so we can all see. Australia there. So to the left and to the west and to the north, there's sort of a little chain of islands called Indonesia in that area. Okay, so that's where we are on the map. Now, when you look at that on a closer up map, what you see is that chain of mountains, you can have a seat if you want. You can quit playing Vanna White for me. You see a chain of mountains that stretches from the Southeast Asian peninsulas all the way into that chain of islands. And you can easily imagine animals and plants migrating through that chain as it developed. No problem with that. So the questions that I'm going to ask you are going to relate to how did they get there, and once they got there, if they're that remote, why are they that different? Okay, so rewatch the video if you want, read the instructions, and some of the questions on the test, and this is one of them, are just what do you think? Can you think critically about where animal and plant species come from and why they are diverse as they are? Okay, so I'm not going to tell you any more than that because I don't want to give you the answer. All right, emergent properties. Do you know what that means? That's a theme we're going to hit again and again and again. So what does it mean to be an emergent property? Okay, so we have these levels of organization, which is the next thing on our list. And what you find is that new characteristics emerge when you combine things at one level into something at a bigger level, okay? So there are some examples of that in the book. I specifically focus on emergent properties of water for a couple of reasons. One, it's such a great example, and two, we actually study the properties of water when we get into chapter 2. So pay attention to that. And here, I'm, again, asking you to think about things and make some connections. So when you get to a question you think, I don't remember that on the review, or I don't remember that being covered in the book, then chances are it's because I want you to think about it, okay, and come up with information. So some of the questions on the test really are designed that way, all right? Most of the questions on the test, you're going to find the answers straight in the reading or straight from the lectures. But there are going to be some that aren't going to be that way. Don't panic when you see those. Sit down, think about them, think them through, and you ought to be fine. Okay, so levels of organization. I like to divide these into 2 categories, those smaller than an individual and those larger than an individual. So if we start at the lowest level, that's atoms and molecules, right? We combine them into cells. If an organism is bigger than a single cell, then we go to tissues, organs, and then finally to an organism. What about the ones bigger than that? What's the next biggest? You skipped one. You're on the right track, though. So if you have just a local group of individuals of the same species that interact and interbreed, what do we call that? That's a population. And then if I talk about the interacting populations of different species, now what do I call that? That's a community. And if I then go bigger than that and pull in the abiotic, the non-living factors like the atmosphere and the climate, what's that? Climate is just one of those factors. Ecosystem is the answer. So you were thinking the right thing, so on the test you got to be able to come up with the right answer. Most of these are going to be multiple choice. So, in fact, I've written all the questions for Chapter 1, and they're all multiple choice. Okay? I think there's one that's true-false, but that's still sort of multiple choice. Sort of, kind of.
A few choices.
Yeah, most of them have 3 or 4 choices, but some of them are kind of matching. So, but they're still written as sort of multiple choice. Okay, energy, nutrients, and interactions. What are we talking about here? Let's see where we are with all this. Okay, let's back up a little bit because I forgot I had pulled in these slides. So here you see a picture of the Foya Mountains. Do you see it?
Yeah, Mount Barron and Mall.
So it's an extreme close-up. So here's what I was saying about the chain of mountains. Follow my line. Here's a chain of mountains. Okay? So you can easily imagine animals and plants moving along that pathway, right? India's up here. A little farther away than that. But where are the Foyas? The Foyas are here. So there's that little preserve. And do you see that that chain of mountains, first of all, it's not as high. And second of all, it's kind of separate from the main one. Okay, so that leads to our understanding of why they are more diverse and more different than the others. Okay, let's see what's on the next slide. All right, so what are we looking at here in terms of emergent properties? Surface tension of water. The surface tension of water is due to its chemical makeup. The molecules make hydrogen bonds with each other. Do y'all remember that? Just lie to me and say yes. Oh, yes, I remember that, and then write it down. I got to remember hydrogen bonds. Surface tension of water is due to the hydrogen bonding between the molecules. So it's not a property of hydrogen. It's not a property of oxygen. It's a property just of what we make out of hydrogen and oxygen. Okay, next. Biodiversity, what do you see here? You see some ducks. Those ducks are all members of the same population. Okay? The plants, do you see that there are different kinds of plants in this picture? There's at least 3 different kinds of plants. If you can see the picture well enough, you can see there are. Are they the same population? But are they part of the same, what's the next higher level? Community. So we have, you can actually talk just about the plant community, or you can talk about the entire community, which would include plants, animals, bacteria, fungi, whatever. And then if I include the water in my picture, now what? That's the ecosystem. Okay? Are we pretty good with that? Wonderful. Okay, next, characteristics of life. Your book goes into just a few of these characteristics, but it talks in particular about the ability to respond to environmental stimuli. So I call that responsiveness. If you're alive, you respond. And I picked this image specifically because the review question asked if you've got sunflowers that turn their little faces towards the sun during the day, that's the characteristic of responsiveness. Did you know plants did that? Did you know flowers did that? That they can actually move? They move slow enough that you would have to stand there a long time to see them. But if you left for work in the morning and they were facing one direction, and when you got home in the afternoon they were facing a different direction, you would know they moved. Okay, but all of that has to do with the overriding characteristic, which is homeostasis. Homeostasis is the ability of organisms to moderate their own internal characteristics in the face of environmental change. And we're a very sophisticated organism, so we do that quite well. So if we had some kind of stimulus that changed us, and the example I have up here is if your blood sugar gets high, what would make your blood sugar go high? Stress. Yeah, but let's do something more fun than stress. Yeah, you're right. But How about if you just eat a donut or a Snickers bar or whatever? Eat some sugar. What's going to happen to your blood sugar level? It goes up. And so your pancreas can sense that change in blood sugar and secrete a hormone called insulin, which you probably heard of. And what insulin does is it tells your cells to suck up all that glucose, that sugar, and brings your sugar level back down. Does that make sense to everybody as an example of homeostasis? So you had some stimulus that made something go up, and your response was to bring it back down. Okay? The ability to respond to environmental change in a way that keeps you kind of staying the same. Homeostasis means your ability to stay the same, stay in your optimal range. Any questions so far? We've barely scratched the surface of chapter 1, so we have a ways to go. I'm just waiting on this to respond. Okay. Let's see if it's going to let me go to the next slide. All right, our next theme in chapter 1 is unity and diversity. What is this a diagram of? What kind of helix? That's a DNA double helix, right? And what do the different colors represent? The pairs, the ATGC pairs that we drew last time. Y'all remember that? Okay, so what has that got to do with unity and diversity? How can you be both unifying and diversifying at the same time? Well, DNA does that quite well. And the way it does it is by having the same sequences and the genes. The gene is going to be the sequence of those AGCT. Okay, so the DNA nucleotide sequences make up the genes, and those genes are highly conserved. What does that mean, to be conserved? Protected, saved. So they're the same. So if you look at the same gene for anything, an enzyme will have a gene that codes for it. The enzyme to make carbon dioxide turn into carbonic acid, the carbonic anhydrase enzyme. You're going to see the same sequence in humans, in chimpanzees, in monkeys, in armadillos. We're all going to have pretty much the same sequence. We have the same genes. And that genetic unity allows us to recognize that we are, in fact, all related to each other. But there are also mutations. So you see, for example, in the gene for the enzyme needed to make vitamin C, that we have that gene, it's broken. In humans, the gene is mutated. So some genes are conserved and other genes are mutated. And that mutation gives rise to the diversity. So diversity, not just in humans, but diversity across all of the kingdoms of life, gives rise to new traits. For example, they recently managed to extract DNA from some old bones, the bones of Neanderthal. That's pretty cool, because those bones are like 10,000 years old, so, or more. And so to get actual DNA samples from that's pretty darn cool. So what they were able to determine is that the gene for blue eye color is a Neanderthal trait, and the gene for red hair apparently is a Neanderthal trait. So it doesn't mean we're all Neanderthals. It just means that we intermarried. The modern humans intermarried with Neanderthals. So genetic diversity, but also genetic unity. That's the point of that. I sure hope this thing is still recording. All right, classification. There are 3 domains. You need to know the term domain. You can think of it as part of your address. So what country do you live in? That's not the same as your state, county, city, or street, right? So domain is the biggest category. Bacteria and archaea are considered both prokaryotic domains. Why? What does prokaryote mean? Pro means before, in this case, and karyo refers to the nucleus of a cell. So prokaryote means these cells evolved before the nucleus did. Okay, so what's the one group that has a nucleus? The eukarya, which gives us the eukaryotes. So you need to know what domain you're in, and you need to know the other two domains. Okay. Back up just a sec. There's something else that's on the review that I didn't put a slide in here for. You need to know your own classification, and you need to know the classifications of the eukaryotes. So there are 4 kingdoms. Kingdoms. What kingdom are we in? Are we plants? Are we fungi? Are you pretty sure we're not fungi? I have some questionable relatives that I'm thinking pretty much like mushrooms, but that's a different kind of a problem. The most primitive is called the Protista. These are things like little one-celled animal-like things and plant-like things that, because they're not multicellular, they're not classified as plants and animals. So they're the most primitive of all. They're also the most diverse. And there's a lot of argument about whether they ought to all be in the same kingdom or not. Right now, we're just kind of lumping them all into a miscellaneous category, not very satisfactory to the people that study those. But for our purposes, good enough. So if I tell you something's a protist, what do you know about it? It's got a cell with a nucleus because it's in the eukarya, but it's probably single-celled. Okay, some of them are kind of colonial or little clusters, but nothing very elaborate. So are we in any of those? No. So what are we? We're in the animal kingdom. So the proper name for that is Animalia. Okay, and then you probably ought to know what you are. How are you classified taxonomically? What's your scientific name? Homo sapiens. And we underline it because it's hard to write in italics. And that indicates that it's in Latin, or Latinized anyway. Okay, so you need to know that you're Homo sapiens. And that's the genus and species of humans. Your book gives you some examples of different plants, different flowering plants. And that's fine. I'm just not going to ask you about plants. I'm more interested in people. Does anybody feel cheated by not having to know the plants? Okay. There are a number of questions on scientific method. And a few of these are going to follow the case of the horseshoe crab. And the horseshoe crab was not covered in the book. So how were you supposed to know you needed to know about horseshoe crabs? You're supposed to look at my review that I posted because I gave you a link about horseshoe crabs. So you need to read the case and you need to understand the basic premise. And the basic premise is this. These horseshoe crabs are doing what's called spawning. And different animals spawn in different ways, but basically what you have is males and females that come up on the beach same time every year, and the females will release the eggs and the males will release the sperm, and they have external fertilization. So there's just all these eggs all over the beach. It's a mess. I got to actually see that once. It was really pretty cool. A lot of the crabs just kind of roll over and die after that, but a lot of them crawl off and go back and come back the next year. Horseshoe crabs are what we refer to as living fossils, because if you look for them in the fossil record, they haven't changed. When they first appeared in the fossil record from millions of years ago, they're still the same. They still look the same. They're very primitive animals. But the connection that I want you to make is between the spawning rungs and the migratory birds. So you have birds that migrate. They come usually south for the winter, and then they go north for the summer. So we have migratory waterfowl, which includes ducks and geese. And guess when they arrive at Chesapeake Bay? Right when the horseshoe crabs are spawning. Isn't that an amazing coincidence? Well, no, it's not a coincidence. So you're going to use your analytical powers of science to figure out how it can be that migratory waterfowl know when to arrive at the Chesapeake Bay. And you're also going to make some predictions about what the effect of global warming may have on migration, because we're already seeing that a lot of birds are migrating north sooner. Why are they doing that? What's happening every year? We're getting warmer earlier in the year. Winters are getting milder. Some birds are not migrating at all, but others are just getting the cue to migrate earlier in the year. So you're going to make some predictions about what happens to the horseshoe crab, migratory waterfowl interaction if the migration timing is different. Okay? So that's all I'm going to say about that. Be sure you look at that review and make sure you're ready to answer some questions about that. Now, there are other terms on scientific method that are covered in the review. You need to know the difference between a dependent variable and an independent variable. You need to know about experimental and control groups. You need to know about making a hypothesis, making a prediction, testing, analyzing the results. Didn't you just do that in lab? Does everybody remember doing that in lab? Yeah? No? Maybe? I thought it looked like fun. So you got to actually put alcohol on the little Daphnias? Is that what you did? Yes. You also did some statistical analyses. And I'm going to ask you to know how to do that on the test. I'm going to give you some data and have you analyze it. So I've got a different case, not Daphnia getting alcohol poisoning. I promise I'm not going to ask you to do variances. Does anybody feel cheated by not having to do variances? Okay, but can you do ranges and means and modes, so forth? Okay. All right, so a little bit of data, a few cases, some application of scientific method. In Chapter 2, what did we do? To me, this is very, very basic, and you should have a good sense of the basic chemistry. Even though I didn't lecture on it, you need to know about protons, neutrons, and electrons. Okay, you need to know where to look for them in an atom. You need to know which ones have mass. You need to know what the electrical charge is on each kind of particle. You get to remember there's a periodic table in the room. I'm okay with you using it. All right? So you're allowed to look up there and look up anything you need. And I will give you perhaps some elements and expect you to figure them out. Okay? Who's this guy? Mendeleev developed the first idea of a periodic table, and I do expect you to remember his name. Do you need to recognize his portrait? No, let's skip that part. Wouldn't hurt you, but you don't have to. Okay? So Mendeleev developed the periodic table, so he's our first scientist that we're learning. Do you remember how ions are formed and how that leads to the formation of ionic bonds? I'm not going back over that today. What's that, Taylor? Losing and gaining electrons. Do you remember how to tell by looking at the periodic table who's going to lose and who's going to gain? Okay, so there's a little bit of memory work involved. So for these guys, you should already know. If you don't already know, you can always look at the periodic table and figure it back out. But here's what you absolutely have to know. You have to know the symbols that go with each name. Okay, so if I ask you about sodium, and you think sodium is S, you're going to miss it. What's S? Sulfur. What's sodium? P. P is potassium. Sodium is Na. What's P? Phosphorus. What's K? Potassium. That I'm telling you, you have to know. Okay? So I'm not going to tell you what the symbols are. You have to remember them. But the rest of that you can figure out. Can you look at the periodic table and figure out how many covalent bonds hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur are going to make? Okay? If you can't, there's the periodic table right there. For the ions, Everything that's in the first column makes an ion a positive 1. Everything in the second column makes an ion a positive 2. Now, when we first went over this, the only negative ion I gave you was chloride. But since then, we've added the hydroxyl ion, OH. So this has more on the list now than it did when we first did it. You just need to be aware of that. All right, any questions about that? All right, do you remember what the polar covalent bonds are? It's such a short list. Can we remember NH, OH, and C double bond O?
Forgot NHOH.
So we added C double bond O later. And for each one, you also need to remember which side is positive and which side is negative. So can we go through that? The bigger atom is always the one that's slightly negative. Hydrogen's the smallest atom there is. So that means the hydrogen's a little bit positive. But what about right in here? Who's bigger here? Look at the periodic table if you don't remember. Oxygen's bigger, so the oxygen is a little bit negative. Or you can just remember that the oxygen's always a little bit negative. Okay, so if it's not on the polar covalent bond list, but it makes covalent bonds, then it must be nonpolar. And on our nonpolar list, we have CH, we have CC, we have CO, we have CN. And then we have a couple of guys down here that I might have mentioned. That's oxygen gas and carbon dioxide gas. And those are nonpolar. What difference does that make? If something's polar, it's hydrophilic or hydrophobic? What is philia? Phil versus phobe. What's a phobia?
A fear something.
It's a fear, repulsion, dislike. Phil means to love, as in philosophy, the love of learning.
Trash.
So hydrophilia, hydrophilic, loves water. Hydrophobic, repels it, hates it. Okay, questions at this point? All right, so on the chemistry of water, what are we seeing here?
That's H2O.
So this is a polar bond, that's a polar bond, that makes the whole molecule polar. A little bit positive, a little bit negative. And what kind of bonds do we have between the water molecules? That would be a hydrogen bond between them. Okay, acids, bases, pH, and buffers, and this is almost to the end of chapter 2. This is a big, kind of heavy part, and I don't want you to get too bogged down in it, but if you can look at this picture and tell me who's the acid and who's the base, then you've got a good start. One of these is an acid and the other is a base. Which one's which?
One by itself is the base, right?
The one by itself is a base. I like that.
That's wrong?
That's a good, no, that's right.
Oh.
The one by itself is a base. That's an acid. What do acids do? Acids can give up, lose, a hydrogen ion. So it has to have a hydrogen ion in order to lose it. If it's already lost its hydrogen ion, it's not an acid anymore. It's the opposite of that, which is a base. If you see the double-headed arrow, then that means it's reversible. It'll go either way, which means it's just as likely to give it up as it is to take it, which makes it a good buffer. Okay? Now, review your notes. We learned a strong acid, hydrochloric acid. We learned a strong base, sodium hydroxide. We learned how to do scientific notation. Do y'all remember doing that? How did we figure out that the pH of water is 7? How did we decide that? So you had scientific notation. You had to know that the pH of 7 is neutral. So review all your notes on those things. Any questions about that? You know those hiccups are going to get recorded into my thing. All right, just saying. All right, chapter 3, this is the organic chemistry, and a lot of this is, to me, it's descriptive, which means you have to remember the descriptions. So you've just got to learn stuff. Basically, you just got to learn it. How did we draw them? We did ball and stick models. We did structural formulas. We learned some functional groups. Are you learning those? Yeah, there's a list of them in your book. So make sure you learn those functional groups. So if I draw a functional group, I'm just going to have a big matching section with a list of the names of the functional groups and then drawings of them. And you have to match them up. Okay, so flashcards are a good way to go with that. Draw the structure on one side, draw the name on the other side. You can just do the one straight out of the book, and you'll be in good shape for that. There's a bunch of them. There's like 10 of them, I think. I can't remember exactly how many.
On the test?
There's 10 in the book. Sounds like 10 is a good number to put on the test. I may not put all of them on there, but I'll put quite a few. Okay? My favorite ones are the ones we needed, though. So for those that we use, those are the most important ones to remember. And so as you're learning the different kinds of carbs and lipids and proteins and nucleic acids, learn the functional groups that we use to build them, because those are the ones that are definitely likely to show up on a test. Okay? So functional groups. We polymerized. What does that mean? What does it mean to make a polymer? What does poly mean? Poly means many. What is mono? Mono is 1. Di is 2. Tri is 3. We don't usually count past 3, but sometimes we do. Okay, so what's a hexose? Hex is 6, like a hexagon. Pentagon is 5-sided, so a pentose is a 5-carbon sugar. A hexose is a 6-carbon sugar. And we're just going to stick with the basics on those. Okay, so recognizing these, and again, this is from the structural formulas. If you can draw them, you can recognize them. But if you can't draw them, at least study the drawings in your book and make sure you can tell them apart. So how would I recognize a monosaccharide? What would I be looking for? One sugar, but what is that one sugar made of? Carbohydrates. First of all, you have to know that's a carbohydrate. And secondly, you need to remember that there's usually 1 carbon for every 2 hydrogens for every 1 oxygen. So a formula, like for glucose, C6H12O6, or for ribose, C5H10O5, that's a good start. But then the next thing to be able to recognize for your monosaccharides is that all of those carbons are connected to each other in a line, although they can form a ring, and that each of those carbons has its own oxygen attached. And generally speaking, one of them is going to be double bonded and the rest are going to be hydroxyls. And then all the rest of our bonds are just hydrogens, okay? So if you see carbons in a row and almost every one of them, or every one of them has an oxygen attached, so that there's 1 oxygen for every carbon, chances are it's a carbohydrate, okay? If I wanted to make a fatty acid instead, what would that look like? First of all, it has to be an acid. So you have to recognize the functional group that is the acid, and that functional group is the carboxyl group. So there's the carboxyl group, okay? Then you have to recognize that to make a fatty acid, the only thing I have left is a bunch of carbons stuck together with nothing but hydrogens on them. They can be double bonded or single bonded, but it's nothing but carbons and hydrogens. And what does this little dotted line mean? It's just more. It's just too long of a line. I got to stick 14 carbons in a row, and I don't feel like drawing the whole thing. Okay, so far? So can you tell the difference between that and a monosaccharide? I mean, to me, they're like, I don't know, easy to tell. Okay? All right, what about cholesterol? What do you know about cholesterol? Structurally, it's a ring. It's actually a complex of rings. And I promise you, I have never memorized the whole structure. I just know what the ring pattern looks like. Every time I want to draw it accurately, I look it up on Google. But does that look anything like what I drew for a monosaccharide or a fatty acid? No, it's 4 rings. And there's like 1 little oxygen. So there might be a little hydroxyl group here. There might be a double bonded oxygen over here, depending on which steroid I'm drawing. But all the rest is nothing but carbon and hydrogen. Okay? And we know those are nonpolar bonds, and therefore it's a lipid. Are you with me so far? In terms of the drawings and being able to recognize the structures. All right, what does an amino acid have? We start with a central carbon. If you're drawing it yourself, this is the way I draw it. I put an amine group on one side and a carboxyl group on the other side. And then I'll have an H group and some kind of an R group. Do y'all remember that we played with R groups a little bit? Yeah, on the test, I'm likely to fill that in. I'm not going to use the term R. First of all, it's not accurate. Second of all, it gives it away. And I'm not in it's not my inclination to give it away. So I might put a specific R group on there. I won't put a big complicated one, but I'll put something on it. Okay, so how do you recognize that this is an amino acid? Okay, so there's your amine, and there's your carboxylic acid group. So you look for those two. Now, beyond that, you also need to be able to put those together into a dipeptide, and we did that in class. And you need to be able to recognize where the peptide bond is. Okay, so if we draw a dipeptide, we start off the same way. But we're going to do a condensation reaction here, and what we're going to wind up with is that the carboxyl carbon is going to bind to the nitrogen of the next amino acid. So here's one amino acid and here's the other. Can you recognize where the peptide bond is? It's between the N and the C that has a double bond O. So that's how to recognize that. Okay, so that's definitely going to be on the test. Okay, and then our last monomer was the nucleotide. Do you remember the parts of the nucleotide?
Pentose, phosphate, nitrogen.
Okay, so we had a pentose-like ribose or deoxyribose, right? We stuck a phosphate on that.
Nitrogen base.
And then we had a nitrogen base attached to that. And there were 4 or 5 different nitrogen bases, right? Do y'all remember that? Do I expect you to be able to recognize the structural formula of those nitrogen bases? Not exactly. I do expect you to remember the difference between a purine and a pyrimidine. And you can remember this by how many rings they have. How many rings does a purine have? Backwards. The bigger word has the smaller structure. So purines have a double ring, and pyrimidines have a single ring. They all have lots of nitrogen in them. And so what's the term for that? Nitrogen base or nitrogenous base. Okay, back up just a sec. So what were the different nitrogenous bases found in the nucleotides? Adenine and guanine. And our pyrimidines? Yep, very good. There are 3 differences that I taught you between DNA and RNA. Can you name them?
DNA has deoxyribose. RNA has ribose. DNA has thymine. RNA has uracil. And, it's the, I can't find the last one. I know it, I just can't find it.
Does anybody else but Josh remembering what we're doing here?
And I don't remember this on my paper.
He's reading it very well. You're allowed to use your notes in class. Double bond, double helix.
Yeah.
What was that? Yes. DNA is double helix. RNA is usually what we call a single strand. RNA is what? Yeah, RNAs tend to fold up. They do. Whereas DNAs, well, DNAs fold, but they fold with help from proteins. So, and we'll worry more about that when we get into what they do for a living. For now, we're pretty much just focusing on their structures.
If I fail
Okay.
this test, can I still pass your class?
What?
If I fail this test, can I still pass your class?
If you fail this test, can you still pass this class? If I say yes, will you keep working or will you give up?
If you say no, I probably won't.
Okay. All I can tell you is there's a direct correlation between how much you understand and study and how well you do on the test. So years of experience, you guys. If you do the reviews, if you answer them and study them, you'll do well on the test. Okay? If you stay up all night the night before you take this test, and I've had people do that, and then you fall asleep during the test, that's probably not a good idea. So it's better to study 3 days ahead, get a good night's sleep, get some exercise, eat well, try not to stress. But I know that's kind of a ridiculous thing to say, but still, okay? Still, studying ahead is better. Sleep consolidates memories. So if you do it the night before and you pull an all-nighter, first of all, you're exhausted. Second of all, things tend to get confused. So try to study one thing at a time. I have to tell you a story, but I'm going to stop the recording first. More than that. If you are not, when you leave my class, if you don't give my class another thought until the next time you come to my class,
I'll think about it. Don't understand it.
okay, you need to find some effective study habits. You do. But if all you're doing is coming to class and that's it, you're not doing your job. Okay? That's like showing up for work but not actually working. So come and see me if you need help. Do it sooner rather than later. All right, functions. We didn't focus on these that much. And I'm not testing you on the nitty-gritty of functions, but there's a little bit on protein functions. You need to know that some of your carbs are structural and some are functional. Some of your lipids, you actually use them to build things in your cells. Some you just burn for fuel. Okay? So when we have